now for me, it's time to introduce our today's speaker, Dr. Muskaan Belani, who has graciously accepted the invitation for this science dialogue series. So, welcome, ma'am. So, for our participants, Dr. Muskaan Belani, she is currently affiliated with the Maharaja Sayaji Rao University of Baroda, where she is presently serving as a research associate in a DBT-Builder CAT 3 project. She has earned her doctorate while working on the cellular and molecular mechanism involved in PCOS patient at the Department of Biochemistry at the Maharaja Sayaji Rao University. She has more than 6 years of experience in the industrial research at the Zydus Cadler Research Center, where she has dedicated for the development of polyclonal antibodies and indigenous diagnostic kits for the analysis of hormones from the urine samples. She has also been involved in the development of different bioassays for the screening of novel chemical entity. So her research expertise is a vast, and currently she is also contributing in the academia for the by teaching the undergraduate and PG student at the Vikram Sarabhai Institute of Cell and Molecular Biology, the MS University of Baroda, more prominently known as CMB MSU. Currently, she is working for the research related to the understanding of the role of exosome from the human adipocyte derived stem cells in ameliorating metabolic disorders. So, this is just a brief introduction about the Dr. Muskan. So, ma'am, we welcome you on this series, and now we expect that our students or the faculties or whosoever is joining us as a participant for this dialogue lecture series, they will be benefited from your research experience, and definitely will get some more insight about the recent research related to the PCOS differential mechanism and its regulation. So we welcome you, ma'am. Now we are handing over this online platform to you so that we can initiate the today's lecture.
Thank you, Poonam ma'am. Thank you, Gopalji, Gopal sir. Thanks a lot for allowing me to present my work at this vast forum. Let me present. Let me start. Okay. Uh, okay. Is the screen visible in the slideshow mode?
Yes, yes, visible.
Okay. So, good evening, everyone. I'm Dr. Muskaan A Belani, and I'm about to present my work on PCOS. It's differential regulation and management. So, one of the most important event in woman's life is to give birth, which is fulfilled with the help of female reproductive system. The system has a pair of ovaries. If the ovaries are fully functional, okay, then these ovaries, they have very important granulosa cells. These somatic cells play a very important role in ovary by helping in steroidogenesis, oocyte growth, and also by undergoing molecular atresia. The granulosa cells are so important in ovary. They have a specialized signaling system in them. So in the signaling system, if we see, the insulin signaling system is present, wherein insulin and IGF, they have vast functions. They help in proliferation and they also help in glucose uptake. Apart from that, the gonadotrophin system takes care of the entire steroidogenesis. So, any insult to these signaling process will ultimately lead to infertility like condition. Infertility means failure to achieve a clinical pregnancy. Now, in 2016, according to WHO, 10 to 15% couples were found to be infertile. And now in 2024, again by WHO, this percentage has increased to 17.5%, indicating that even though there is vast and vast research going in this field, the percent of infertile couples is increasing. We will come to know why the situation is like this. So the causes of this infertility are, they can be genetic, they can be female oriented with respect to ovary. They can be male oriented with respect to the male factor problem. They can be at the level of tubal infertility. And sometimes it goes idiopathic. The causes are unknown. So in the present scenario, a female, she is exposed to the change in lifestyle. There are a lot of changes in eating habits also. So these things together lead to the condition like insulin resistance. Insulin resistance is the primary cause for amenorrhea, very less amount of very less menstrual cycle, poor fetal outcomes, and also miscarriages. Insulin resistance also leads to the condition like PCOS, that is polycystic ovarian syndrome. This is detected ultrasonography by the presence of cysts. Here is the picture, lots of cysts. Along with this, the patient also shows hyperandrogenism, lots of androgen levels, chronic anovulation. Now, this insulin resistance like condition is present or can be said is a pathogenesis of 60 to 70% of the PCOS cases. But rest of the 20% PCO cases are without insulin resistance, which we should take care. Not all the PCOS cases will be having insulin resistance in them. Amongst the 60 to 70% of the PCOS cases, 70 to 80% of them are obese, that is BMI greater than 25, and rest are lean PCOS, 20-25%. So in insulin resistance also there is obese and lean, and amongst PCOS we have non-insulin resistance patient also. So if we see signaling in the PCOS granulosa cell specifically, so because of insulin resistance condition, there is resistance at the metabolic activity. But what about the steroidogenic mechanism? That thing is not known. Apart from that, what about the signaling in PCOS non-insulin resistance condition? So if it is non-insulin resistance, that means insulin signaling should be functional. And what about steroidogenesis? So the changes or the pathways are still not known. So we thought of unraveling these mechanisms, what is happening at the level of steroidogenesis in IR and NIR cases, and what is happening at the insulin signaling. So we designed an experiment in which we availed the follicular fluid samples from the infertility clinic. Isolation of cells was done, and after isolating the cells were characterized for the presence of granulosa cells with certain markers. From the hospital, when we availed the samples, we were getting samples as control and as PCOS. So those PCOS samples were not distinguished for insulin resistance condition. So at our lab, we distinguished the cells for being insulin resistance by the help of protein expression of insulin receptor beta, which explained and which helped us to segregate the PCOS samples as insulin resistance and non-insulin resistant. So we got three groups then: control, PCOS insulin resistant, and PCOS non-insulin resistant. And further, we tried to find the protein expression of insulin and steroidogenic signaling, mRNA expression of the IGF system and fat metabolism, and hormone analysis of the uh, granulosa cells. Okay, so this is the slide explaining the characterization of cells by immunocytochemistry, wherein we observed the cells to be positive for CYP19A1, also known as aromatase, which is the marker for granulosa cells. Apart from that, we also we also observed them to be positive for 3 beta HSD as well as 17 beta HSD. All the three are the markers for steroidogenesis, which is very important in the granulosa cell. So this confirmed that the isolated cells were granulosa cells. Further, as I said, we wanted to see whether they are insulin resistant or not. So in that condition, we observed for the protein expression of insulin receptor beta in the cells. So here we observed reduced expression of insulin receptor beta in some samples of PCOS as compared to the rest. Those samples were segregated as insulin resistant and rest were segregated as non-insulin resistant. The significance was quite significant. The decrease was quite significant as compared to control and non-insulin resistant. Moreover, when we calculated the number of patients which we found to be having decrease in insulin receptor beta, 70% of the patients were having IR were found to be PCOS IR as compared to the non-IR other patient, which was as compared to the literature explaining that 60 to 80% of the patients are insulin resistant in PCOS. These cells were checked for the person's cell viability by trap and blue method, and here we observed significant decrease in the cell viability in PCOS IR as compared to control. Decrease was also observed in the non-insulin resistant PCOS cells. However, the decrease was less significant as compared to IR, which was attributed to the down regulation of insulin receptor beta, which otherwise is helpful in survival and proliferation. Now, hyperinsulinemia usually accompanies insulin resistance condition because insulin is not able to be utilized by the cell, and so there is increased amount of insulin. So what is happening beyond hyperinsulinemia? What is happening to the parameters, the other signaling molecules? So we ought to understand the regulation of IRS1, PI3 kinase, P38 MAP kinase, ERK1/2, protein kinase C, PPAR gamma. So we tried to understand. We observed that there was decrease in the expression of PI3 kinase, phospho-AKT, protein kinase C in the insulin resistant group only, whereas there was no change in non-insulin resistant group. And there was an increase in phospho-IRS, and this phosphorus was serine 307, which indicates that there is an insulin resistance condition. Instead of tyrosine, phosphorylation is at serine 307. So this entire thing, the entire PI3 kinase, phospho-AKT, protein kinase C, eta downregulation indicated that translocation might not be taking place on the cell membrane, because of which glucose uptake is not possible. And so glucose uptake is not possible, and so there is the condition of insulin resistance. And so this condition is not seen in the non-insulin resistance PCOS patients. They are same as the control group. Other than that, we observed that there was increase in the expression of ERK1/2 and P38 MAP kinase, and the increase was observed significantly in both the groups, IR as well as NIR. Okay, this was attributed to the increase in oxidative stress. Other factors like osmotic stress and other conditions, they might be leading to the condition like PCOS as a whole, because of which we observed increase in the ERK1/2 and P38 MAP kinase. We also observed increase in PPAR gamma significantly in both the groups. Now, this PPAR gamma, also known as peroxisome proliferator activated receptor, is responsible for the matureness of the follicle, responsible for the matureness or differentiation of the follicle. Increase in PPAR gamma leads to arrest of the follicle, and hence the increase was observed. So this increase indicated that there is an increase in the inflammatory response in PCOS condition, and because of PPAR gamma, there is increase in the cyst formation. Apart from the insulin signaling pathway, we then wanted to understand what is happening at the fat metabolism in granulosa cells. So SREBP1C, FAS, ACC1, CPT1, these are the genes which are responsible for the uptake of cholesterol and helping them to transfer to oocyte for its growth and also for the purpose of steroidogenesis. So when we analyzed the gene expression of these molecules in both the groups, observation was that significant increase was observed in the insulin resistance group as compared to the non-IR group. Again, because of the hyperinsulinemic condition, there is an increase in SREBP1C and FAS, ACC and CPT1. The same is not observed in NIR. Along with insulin signaling, IGF system is also known to play an important role in granulosa cell, from the conversion to the pre-antral follicle to the Graafian follicle. The IGF system entire is important. Moreover, in the IGF system, IGF-1 and 2 subunits are there, amongst which IGF-2 plays an important role in the Graafian follicle formation. Apart from that, the receptors are also present, IGF-1 receptor and IGF-2 receptor. IGF-2 receptor is also known as the decoy receptor. So here, we observed that there was a decrease in the IGF peptide only in the PCOS IR group as compared to the non-IR group, and increase in IGF-1 receptor in both in only the PCOS IR group. This was again attributed to the insulin resistance condition that is prevalent over there. And upregulation of IGF-1 receptor ultimately stimulates differentiation to the luteal phases and ultimately to the cyst formation. IGF-2 peptide, as I told you, plays an important role in the PCOS condition, and so we observed an increase in that. IGF-2 receptor being a decoy receptor, its increase was observed in both the condition. So upregulation of IGF-2 receptor, it ultimately sequesters the IGF-2 and limits the activation of IGF-1 receptor and IGF insulin receptor beta, thus inhibiting the growth. This ultimately inhibits the growth of the granulosa cells. Having understood the insulin signaling mechanism, the fat metabolism genes, the IGF-1 pathway, we now wanted to understand how steroidogenesis is playing a major role, how insulin resistance is affecting steroidogenesis. So, steroidogenesis takes place in granulosa cells, wherein they take the androstenedione and testosterone from theca cells. The delta 5 pathway is active in theca cells, whereas delta 4 pathway is active in granulosa cells. In the granulosa cells, there is a STAR, steroidogenic acute regulatory protein, which helps in the uptake of cholesterol from the outer membrane to the inner membrane of the mitochondria. Then we have CYP11A, also known as P450SC side chain cleavage enzyme. This is the key regulatory enzyme of steroidogenesis along with aromatase. And 3-beta-HSD, 17-beta-HSD will help in production of the hormones progesterone and estradiol. We wanted to understand the what is the expression of these genes and proteins in the PCOS condition. So the real-time analysis of these genes explained that downregulation of the entire steroidogenesis was observed in both insulin resistance as well as non-insulin resistance condition. Okay, now up till now we have observed that in insulin signaling, fat metabolism and IGF-1, only the IR cells are getting affected, whereas in steroidogenesis, both the groups, IR as well as NIR, both are getting affected. The same thing was observed in the protein expression, that 3-beta, 19-beta, 17-beta, all were getting down-regulated in the protein expression. Whereas the gonadotrophins were showing an increase, FSHR and LHR, they were showing an increase, which is again attributed to the literature, wherein in PCOS condition, there is surge of LH, and so these gonadotrophins show an increase in the cells. So having observed this, we then further analyzed for the hormones, progesterone and testosterone. There was a decrease in progesterone in both the conditions, IR and NIR, which is again attributed to the environment of the cell. Because of hyperinsulinemia, there is excess of secretion of other factors like GDF9 and BMP, that is growth differentiation factor 9 and bone morphogenetic protein 15, which inhibit progesterone production in granulocytes. Actually, progesterone is a very important hormone for the stability of pregnancy, but because this hormone is going down, actually CYP19-1 FVC progesterone hormone, this is after beyond the CYP11A. CYP11A is increasing, but ultimately because of some defect over here, we observed that there is a decrease in the progesterone hormone. To our surprise, testosterone hormone showed a significant increase in the insulin resistant patient as compared to the non-insulin resistance. This was a great marker to segregate PCOS IR from NIR. Now, testosterone is increasing. Why? Because there is piling up of the androgens, because CYP19A1 is not working. CYP19A1 is not working. This also explains the fact that PCOS, as we told, are of two types, insulin resistance and non-insulin resistance. So, the cyst observed is there in both the cases, IR as well as NIR. You will find cysts in both the cases, but when it comes to the hormone levels, there is piling up of androgens in the insulin resistance condition because of hyperinsulinemia, whereas the same thing is not observed in the non-insulin resistant part. So this is only polycystic ovaries, whereas this is polycystic ovarian syndrome patient. So having analyzed all these things, we observed that when we compare a normal granulosa cell to an IR and a non-IR, then there is an increase in the receptor in both the condition. Steroidogenesis is decreasing in both the condition. Whereas in insulin resistant condition, the insulin signaling is affected and steroidogenic signaling is affected. And these parameters are not at all, insulin signaling is not at all affected in the non-insulin resistant patient. Steroidogenesis is affected in both the cases, whereas PPAR gamma and the growth factors are getting affected in both the cases. So having differentiated, so this was about the differentiation, the differential regulation of PCOS IR and PCOS non-IR. We could differentiate this. So these candidate molecules, these can help us further to differentiate between the IR and non-IR conditions. We can also choose these target molecules to design certain therapy or diagnostic tools to differentiate between these two conditions. Actually, today also at clinical level, PCOS patients are not being segregated as IR and IR. This is at a clinical level. At the molecular level, we have done it. In clinical, hardly any patient is tested for insulin signaling. They're not being tested for insulin resistance. The doctors are directly observing in the CT scan, in the sonography report, that the cysts present are there or not, and they are depending upon the androgen levels, and then they are treating them. So this research is in that way contributing to specified therapy for IR as well as NIR patients. With this, actually, we have a publication in the Journal of Steroid Metabolism, but I could not bring the picture over here. Now, coming to the other part of the story, once we observed that the candidate molecules are differentially expressing and they can aid in designing target therapy, we ought of understanding whether any herbal molecule can help to differentially treat these two conditions. If we see today's market, then PCOS patients, they are treated with thiazolidinones, metformin, dicairoinositol, spironolactone. These drugs are available in the market, but these have one or the other side effects like fetal growth restriction, weight gain, then no ovulation rate has been observed in some cases. In some cases, there are GI tract problems. Malabsorption of vitamin B12 has been observed, lactic acidosis, lot of other effects, and they are not recommended. Metformin actually is not recommended for the normoinsulinemic, that is lean patients. It is not recommended. So none of these ISDs have increased live birth rate till date. They don't have. So we thought that can we think of any novel bioactive to ameliorate these two conditions, these two phenotypes of the PCOS differently? We just thought about if we can. So our lab already had worked on Swarsha marine, which is a herbal compound, and had seen its effect that it is hypoglycemic, hypolipidemic, anti-inflammatory, insulin sensitizing potential, and also it was observed to have a good potent insulin sensitizer in the STZ-NA diabetic rat models. So we thought of using this herbal drug to see its effect on the two different conditions, two phenotypes of PCOS, that is insulin resistance and non-insulin resistance. And we took metformin as the positive control, which is already known in the market and has been given to the patients of the PCOS. So when we used metformin in our experiments, we first saw the person cell viability. So as earlier seen, PCOS patient, there was decrease in cell viability in IR as well as NIR cases. So both swarshamarine and metformin were able to revert back the cells and revert back the condition, and the person cell viability was observed to be good in both the conditions. However, in NIR condition, metformin helped in proliferation of the cells also. Proceeding further, we analyzed for the herbal compound and metformin together on the insulin signaling proteins. So here, the blue one explains that this is for the PCOS IR condition in this slide. So sorchamarine and metformin both differently were observed to revert back the insulin resistance. The insulin receptor beta was increased. There was decrease in the phosphorylation of IRS, increase in PI3 kinase, AKT, protein kinase C, and the oxidative stress level, which was determined by MAP kinase P38 and P4442, was also found to be significantly reduced, and PPAR gamma levels were also significantly reduced. So this indicates that sorchamarine and metformin both were potent enough to show their significance on the insulin resistance condition. Now, what about the non-insulin resistance condition? As seen in the non-insulin resistance condition, insulin, none of the metabolic parameters did not show any effect. So even sorchamarine did not show any effect over here. But to our surprise, metformin reverted back the basal levels also, insulin receptor beta, then PI3 kinase, phospho-IRase, phosphocate. So metformin showed its effect beyond the basal level, whereas at the P38 MAP kinase in P44, the oxidative stress level, both of them were equally potent. They reduced the oxidative stress level in non-IR condition, and they were found to be equally potent. PPAR gamma, in case of PPAR gamma, only metformin was found to be significantly reduced. It reduced the PPAR gamma. So this indicated that sorchamarine had no role to play in non-insulin resistant condition, no role to play on insulin signaling, but metformin showed its level at activity beyond the basal level, indicating that it, if given to those PCOS patients who are not insulin resistant, then it might show some toxic effects. It might show some toxic effects. Sorry. Yeah. So metformin was observed to decrease the basal insulin levels in normoinsulinemic subjects, demonstrating its toxic effects. Further, we wanted to observe for the effect of the bioactives on the fat metabolic genes. So in the insulin resistance condition, sorchamarine and metformin were found to be potent enough to bring back the elevated levels of the fat metabolic genes, whereas in the non-insulin resistant condition, sorchamarine actually did not show any change, whereas, and as such, we saw that the fat metabolism genes, they are not affected in the non-insulin resistance condition. But again, metformin, it did show some decrease over there, some decrease in all the parameters. We then proceeded for the levels of the steroidogenesis. We then proceeded for the levels of steroidogenesis, that is PCOS IR in the non-IR condition. So, as observed earlier, in PCOS insulin resistance condition, star, CYP11A, 3 beta, CYP19, 17 beta had shown some decrease in IR condition. So, that decrease was reverted back by sorchamarine and metformin. In CYP11A, they decreased the increased activity of CYP11A1 in IR condition. In NIR, steroidogenesis was observed to be affected in both the conditions, insulin resistance as well as non-insulin resistance condition. So herein, when sorchamarine and metformin were added, sorchamarine and metformin, they showed reverted back star 3 beta, CYP19, 17 beta. However, metformin showed a better effect. Metformin showed a good effect or beyond that. It brought back the steroidogenesis to normal. But sorchamarine was unable to do that. Sorchamarine was unable to do here. Sorchamarine and metformin were found to be potent in insulin resistance condition equally, whereas in the non-insulin resistance condition, only metformin was found to be doing its job of reverting back, but not sorchamarine. Apart from after the gene level, we observed for the protein level what is happening at the steroidogenic levels, steroidogenic procedure. So here again we observed that Swarshamarine was able to, and metformin were able to revert back the protein levels in the insulin resistance condition, whereas only metformin was able to revert back, but Swarshamarine did not show any effect in the non-insulin resistance condition. So, to summarize the entire thing, when Swarshamarine and metformin are added differently to the PCOI, to the polycystic ovarian syndrome insulin resistant patients, IR patients in the IR granulosa cell, then Swarshamarine is able to restore the insulin sensitivity and steroidogenesis. Similarly, for metformin, it is also able to restore, and both are equally potent. They are able to bring back the PCOS insulin resistant back to the normal granulosa cell. But when observed for the non-insulin resistant patients, observed for the non-insulin resistant granulosa cell, Swarshamarine is not able to do its job. Actually, insulin resistance condition is not prevailing. It is at the basal normal level. So Swarshamarine is showing no change over there. It's no showing no change over there. But metformin, however, it is restoring the steroidogenesis, unlike Swarshamarine, which is not doing anything at the level of steroidogenesis. But apart from that, metformin is also showing its toxic effect by increasing the basal insulin sensitivity. By increasing the, so it might, if given to the patients, it might bring that patients to lot of loss of weight, So to conclude, a proper diagnosis of the insulin resistance condition in PCOS is required so that targeted therapy can be prescribed to achieve increased pregnancy rates with decreased time. So if the doctors, they target the patients and they go for their personalized treatment, then it will be beneficial to the patients, because targeted therapy can be given to insulin resistant condition as compared to the non insulin resistant condition, and clinical pregnancy rates can be increased with this. So thank you, sir, thank you, ma'am.